So our speaker today is just down the road in Notre Dame, but she wasn't able to attend here today because she's doing some really sensitive experiments. And just in case things go wrong, she needs to be there. So she still agreed to do this presentation, I'm assuming from your office.
Yes.
At Notre Dame. Her name is Amy Hixon. Everyone should have received bio and information about her. So I'm just going to go into, how many of you know where the actinides are? Actinides? What do you know about the actinides? Huh? What do you know about the actinides? They all what? All synthetic? Not all of them, but the majority. What else you know? One person speak up loud so I can hear. I'm hearing multiple voices, but yep, I think most of them are radioactive. Okay, so that's the area of chemistry we'll be looking at today. Now, the other question that I want you to pay attention to in Dr. Hixon's presentation is the fact that her research is being funded by the US government. Why is the US government interested in this last row of elements? Weapons, in particular, what kind of weapons? Nuclear weapons, okay? So some of you already have an idea of where this research topic is going to go. So we want to thank you, Professor Hixon, very much for coming here. I think everyone is perked up to hear what you have to say.
Good. Thank you so much for that introduction. So today I'm going to talk to you guys about chemical detection and risk management of the actinide elements. And I'm again here talking to you from my office at the University of Notre Dame, and I'm a faculty member in the Department of Civil and Environmental Engineering and Earth Sciences. And no, that's not the longest department name on campus. I guess we're having a little bit of a competition with the math department on that one. See if I can get this to move. There we go. So just an overview of sort of the risk management side of things, or what people are usually worried about when they hear actinide elements. The ones you might be most familiar with would be uranium and plutonium. Those tend to be the ones that ring bells for people the most often. And so we want to consider the risk to us from both background radiation as well as man-made radiation. And a little bit later I have a slide that sort of talks about the various sources of background radiation, but here we just have a picture overlaying the background radiation over the US. So you can see the areas that have like this pinkish red color have higher background radiation levels than other parts of the US. Here in the Michiana area along Lake Michigan, we have a fairly low background radiation dose. And then people are also worried about man-made radiation. So as Professor Murray mentioned in the beginning, or perhaps one of you from the audience stated, nuclear weapons are a concern of everybody, but also nuclear power and nuclear medicine tend to be at the top of people's lists. So I wanted to sort of start off with just a little bit of terminology. I call these the 4 R's, and they're commonly misused, especially in the media. So I like to sort of get this out there as much as possible. It's like my little bit of outreach. So I like to think of radiation and radioactive decay and all of these things with the analogy of a light bulb. So radiation refers to emissions from the actual decay process. So for our light bulb, that would be the light that is emitted, the light that you see. Radioactive decay refers to the spontaneous change of an unstable or excited nucleus to a more stable form, accompanied by the release of radiation. So this is what's going on in the light bulb that leads to the process of producing light. Radioactive material refers to a collection of unstable or excited nuclei, so that would be the light bulb itself. And then radioactivity itself is more of a catch-all term, so this can refer to the process of radioactive decay or radioactive material. So try to keep those in mind as I go throughout the rest of my talk today. So why are nuclides radioactive? Well, it all boils down to the fact that their N/Z ratio, or the ratio of neutrons to protons, is either too high or too low. And so the nucleus is unstable and does something about it. So if we have too many neutrons, then the nucleus will change a neutron into a proton and it will emit what we call a beta minus particle. If we have too many protons, then the nucleus will change a proton into a neutron, and we have what's called beta plus decay. And if we have too many nucleons just in general, then the nucleus will emit both a proton and a neutron. So it essentially will emit a helium nucleus, which we refer to as alpha decay. And so each of these different particles behaves differently in the environment. And so this figure here is just showing how the difference between alpha, beta, and gamma particle, or gamma rays, excuse me, how they're attenuated or stopped, and how we shield ourselves when we're working with different nuclides that have these properties. So alpha emitters, most of the actinide series are pure alpha emitters. They're relatively weak particles, and they can be stopped very easily by a piece of paper, or even by the dead layer of skin cells that you have. So elements that emit alpha particles only aren't a really large health problem unless you ingest them or inhale them. So, you know, I'm not very fond of drinking my solutions. Usually that's against a lab policy. So there's not a huge health hazard to me working with these particles in my laboratory. Beta particles, they can move a little bit further. They're a little bit more, they interact more with matter. So we need something more like plexiglass or some wood to stop it. Whereas gamma rays, which yeah, they travel the furthest, and you need something like concrete or water to attenuate them and protect yourself. So that's why a lot of nuclear reactors are made out of some special concrete, and the reason that a nuclear core is suspended in water is basically to protect those people working at the nuclear power plant from gamma radiation, and also as a way of attenuating temperature heat from the reactor. Another thing that's really a big point of contention even nowadays, even after all the time that we've known about radiation and radioactivity, is what is the radiation effect to humans? So here we have an arbitrary scale of the effect that radiation has to humans versus the dose in a unit we call REM. So way down here is the typical dose that you or I or any member of the public would receive from background radiation. This green dot here represents the annual exposure limit to a member of the public. So what that means is this is the largest amount of dose that you're allowed to receive from, say, a nuclear power plant or most of the nuclear medicine practices. We increase that by about a factor of 5 for people who work with radiation and radioactivity on a regular basis. And then, you know, double that is the lowest dose that's ever been linked to an increase in cancer. So we have 3 different theories here that describe how radiation produces health effects in humans. So the first one is called the linear no thresh effects theory, which is represented by this red line here. And this says that no matter how much dose you have, there will be an effect. Whether or not, you know, it's a 1-to-1 ratio is up for debate. But the point being that no matter how little radioactivity you're exposed to, there will be a health effect manifested. Conversely, we have the threshold effect theory, which is this blue line. So we basically say there is no effect until you reach a certain threshold, and then you're going to start seeing human health effects. And right now this graph is drawn this way, where the threshold effect starts at 10, just because that's where the lowest dose has ever been observed or linked. But most experts think that it's actually much higher than that. And then there's also this green line, which is called the hormesis theory. And this basically says that, hey, maybe a little bit of radiation is actually beneficial. And this can be, you can think about this, you might have heard in the news that perhaps drinking a glass of red wine might actually be good for cardiovascular health as opposed to having detrimental bodily effects. So this is, something along those lines of having low doses might actually be beneficial as opposed to being harmful. And I'll just point out that way up here at 50 rem is where we consider changes in blood chemistry and acute effects or radiation sickness or radiation poisoning to occur. So here's that slide I mentioned sort of at the beginning, talking about different background radiation sources, which we can divide up into natural radiation and artificial radiation. So natural radiation would be from food and drink. I don't know if you know, but bananas are actually radioactive because of the potassium-40 that they have in them. You get some exposure to radiation just from being in buildings or being outside. Granite that's used in a lot of buildings has uranium and thorium in it, which are both actinide elements. We have radiation effects, or we are exposed to radiation just for being part of the universe. So if you recall back to that map of the US I showed at the beginning, if you remember, there was a higher background radiation for a lot of the places in the West along the Rocky Mountains. That's just simply because those people are higher up in altitude, so they're going to have a greater background radiation. Radon is a radioactive gas that is produced from uranium-bearing minerals. A lot of times when you're buying a house for the first time, if there's a basement, they recommend that you do a test for radon to make sure you're not accidentally poisoning yourself. And then as far as the artificial radiation goes, we have nuclear medicine. So X-rays, if you've ever had a PET scan, or sometimes for MRIs, they use different contrast agents. A lot of those have radioactive components. And then finally, and I'll really drive this point home, only 1% of your entire background radiation comes from the nuclear industry. A lot of people are super afraid of being located near a nuclear power plant, where that's really not where a lot of their dose is coming from. The industry is very heavily regulated, and as I showed on the graph before, the amount of radiation exposure that we're allowed to give people is rather low. So this is probably the most crowded slide I've ever shown in a presentation ever, and I apologize for that. But I really like this slide because it sort of shows everything all at once as far as dose effects goes. So each of these black lines shows the dose itself. So we're going from 0 to 1 millisievert. On the bottom, this is 0 to 10 millisieverts, 0 to 100 millisieverts, 0 to 1,000, 0 to 10 sieverts, and 0 to 100 sieverts. So these little green arrows that you see all over the place, these refer to the background levels in a lot of places. So this is right where we are in the US. Most of our background radiation is about 3 millisieverts per year. And the regulations from our government are, an order of magnitude less than that. So the drinking limit for drinking water systems set by the EPA is way down here at 0.4 millisieverts. We have, if you're going to decommission or break down a nuclear reactor or any kind of other part of the nuclear fuel cycle, you need to make sure that the background radiation or the radiation from that area is way down here at 0.25. Here is again that dose limit to the public, just in different units this time. And as you go up in dose, really what you're seeing is that's when we start seeing uses for, treating cancer and getting into acute radiation syndromes and sicknesses. So at the beginning, Professor Murray asked for reasons why the government might be interested in the actinide materials or actinide elements. And somebody suggested nuclear weapons, and that's certainly true. I actually haven't included that in my presentation today, even though that's a large portion of my funding. But originally they were interested in the actinide elements because of nuclear power and what comes out when we're done with nuclear power. So they use nuclear fuel rods, and how are we going to dispose of those long term? There's a worldwide consensus, so that will be with a geologic repository, which is just a fancy phrase for saying we're going to bury it really deep underground. And so we're interested in understanding how that affects people, especially at very long time periods in the future. So just to reiterate, here's our periodic table, and the actinide elements are the very, very bottom row in the F block, going from actinium down to out to lawrencium. Me specifically, I pulled these three out: neptunium, plutonium, and americium. These tend to be the three elements that I work with the most, with plutonium being my specialty. And this is because when we're talking about long-term toxicity from used nuclear fuel, the actinide elements have the greatest contribution. So I don't know if it's cutting off for you guys here, but the x-axis here is years, and this is relative toxicity relative to natural uranium ore. So you can see fission products, while are really important sort of near term, once we get out to longer time periods, they drop off rather rapidly. And fission products include the lanthanide series, so that's this other the other part of the F block, and also include things like strontium and cesium. Oh. And then the actinide elements, as you can see here, they persist out to really, really super long time periods. So that's why we're really, really interested in their behavior. So I consider or describe myself as an actinide geochemist. So why is that important, or why do we need to understand actinide geochemistry? So we want to be able to treat and dispose of our actinide-bearing waste. And I don't know if any of you have ever heard of the Hanford site or know anything about that, but that was part of the Manhattan Project when we were trying to build nuclear weapons. And a lot of the nuclear waste got put in these big steel drums, and they're now buried. So we're trying to figure out what to do with that now. We call that legacy waste. We want to evaluate the risk of these actinides to human health. And so we have what's called performance assessment, and that's what this part of the figure is showing. So we have some scenario where we have a production facility that's leaking, or a tank that's buried, like the Hanford tanks that's leaking, or something that's called a crib, which is literally a wood-lined trench that waste might have been buried in during the Manhattan Project. We can assume that these are leaking into the vadose zone, the vadose zone being the shallow root zone of the crust, and understanding what factors affect transport from, say, the tank down to the groundwater, and then how fast is it going to move, from here to there, and then how fast or how long will it take groundwater to reach a receptor. So that would be you and me, to reach our drinking water source, or perhaps a river or a lake where we might be fishing or using water to irrigate our crops, or what have you. So that's a lot of the research that's going on, in addition to trying to remediate these legacy waste sites. So why do I care about plutonium specifically? Well, in the United States, nuclear weapons production and testing has led to approximately 10 to the 5th curies of plutonium as subsurface contamination. at the Nevada test site. That's what's shown here. And each of these little sort of pock marks that you see represents a different nuclear test that we performed. And then I mentioned the Hanford site. There's more than 10 to the 4th curies of plutonium-239. The Savannah River site, which is in South Carolina, has plutonium that's buried as low-level waste. So there is plutonium that exists in the environment, primarily from our nuclear weapons testing programs. But it's also a worldwide problem. Countries such as the United Kingdom and the former USSR also are facing these sorts of problems. And it's a problem because plutonium poses a long-term environmental concern because it's not only chemically toxic, but it is radioactive, and it has a really, really long half-life. So for example, this isotope, plutonium-239, has a half-life of more than 20,000 years. And what that means is it will take more than 100,000 years for all of that plutonium to be considered gone or to decay away. So like I said, it's a pretty big problem that we're facing. And just to put, I know these units might be a little different for you or new for you. 10 to the 5th curies is a lot. I work in my lab with something like millicuries is the upper limit of what I'm working with in my lab. So we're talking about some significant levels of contamination here. So what happens to plutonium and other actinides once they've been introduced to the environment? This figure is one I love that was put out by Los Alamos, showing all of the interactions that can occur or that we need to consider. So we can have complexation in the aqueous phase. So, plutonium and neptunium and all the other actinide elements interact with water to become hydrolyzed. They can complex with carbonate is a particularly interesting one that we have to worry about, or, natural organic matter or other organic species that are in the water column. So we need to consider aqueous complexation. We need to consider redox properties. So we can change oxidation state, and how does oxidation state affect the behavior of the elements? We need to worry about bioavailability. So when we're talking about the environment, we're not just looking at, what the groundwater and the soil that the groundwater is moving through, but there's going to be microbes and other things that we need to worry about. And microbes can actually actively seek out plutonium and some of the other actinide elements when they're trying to seek iron. So they kind of, I like to say they mistake it, but it's not like they have brains. But they will wind up scavenging some of the actinide elements instead of iron or other nutrients that they need, and metabolizing them. And so that affects how plutonium and the actinide elements move in the environment. We can talk about sorption, which is what I tend to focus on in my research. So this is how does the actinide element interact with the mineral surface? So we can have direct complexation with the surface. We can have precipitation onto the surface. We can have incorporation into the bulk matrix. There's a lot of different mechanisms we need to consider there. And then just general dissolution and precipitation processes. So all of these ultimately are what we need to take into consideration before we even think about transporting or how they migrate through the subsurface environment. So I figured it'd be prudent to give you sort of a crash course in plutonium aqueous chemistry. So plutonium has a very rich and complex chemistry. That's part of the reason I really love it. It's extremely challenging to work with, but also extremely rewarding. It can be found in 4 different oxidation states simultaneously. So it can be found as plutonium-3, plutonium-4, plutonium-5, and plutonium-6. So here we show what plutonium-3 and 4 look like in the aqueous phase, and they just exist as hydrated ions and retain their overall formal charge. But plutonium-5 and 6, we have these axial double oxygen bonds that form to give us this linear dioxocation, and this lowers their effective charge. So sometimes we use other actinide elements as stable oxidation state analogs. So we might use neptunium, which is usually in the +5 oxidation state, or uranium, which is usually in the +6 oxidation state. And based on those, we would think that the effective charges of plutonium 5 and 6 would be about 2.2 and 3.3. But we've actually measured the effective charge of plutonium-6 as being even lower, about 2.9. So you can see those values here for their effective charges. So the strength of plutonium complexation constants with oxidation state follows the trend that you see here, which we call the actinide trend. And this is one of those things that when I'm teaching my classes, I make sure my students, highlight and circle and star this. It's one of the main tenets of actinide chemistry is this trend here with oxidation state. So we see the trend corresponds to an overall decrease in effective charge. And what that means is that we're going to get stronger complex formation with molecules that have a higher effective charge than those with lower effective charge. So because plutonium-4 has the strongest effective charge, it's going to be generally assumed to be removed from the aqueous environment. So it's going to want to bond with mineral surfaces and things like that. So we assume that plutonium-4 is extremely immobile in the environment. Sort of on the other side of the spectrum, the plutonium-5 ion has this lower effective charge and therefore is happier to be just in its charged state, and is therefore considered to be the most mobile due to its weak complex formation. So as you can see in this table here, the redox potentials for all of the oxidation states has to do with why we can see all 4 of them simultaneously. So under acidic conditions, you can see that the redox potentials are all about 1 volt, regardless of what the actual redox reaction is. And so that just means that even really small changes in the redox potential of the system could result in large changes in the oxidation state distribution. If we raise the pH of our system to about neutral, the plutonium-3-4 couple becomes negative, and we generally assume that plutonium-3 becomes unstable. But the other redox potentials are still pretty close to each other, so you could still have plutonium-4, 5, and 6 simultaneously existing. And then if we raise our pH even further to alkaline conditions, the 4-5 couple also becomes negative, and the other redox potentials aren't as close together. So we usually have plutonium-5 for plutonium-6 in the solutions. I also wanted to point out, if you're looking at just the redox reactions, say going from plutonium-3 to plutonium-4 or plutonium-5 to plutonium-6, you see that there is no dependence, there is no proton involved in those reactions, and that's because you don't have to form or break any of these axial oxygen bonds. You're moving between the oxidation states, where you either have those axial oxygen bonds or they're absent. Whereas, say if you're going from plutonium-4 to plutonium-6, you need to form those the axial bonds, and that's why you need the proton. And those reactions tend to be a lot slower because you need time in order to form or break those axial oxygen bonds. Okay, so let's talk about some real-world scenarios or real-world studies that I've been involved with in the past. So the first little tidbit I'm going to talk about are a project called the Lysimeter Project at SRNL, and SRNL is the Savannah River National Laboratory, and this is in South Carolina. And so what the scientists did is they built these lysimeters. And so this is just, a lysimeter is a fancy word for, it's basically a jug that has the bottom cut off of it and has been buried underground is essentially what it is. So the jug was filled halfway up with sediment from the area, and then they put a filter paper in the middle that had a different oxidation state of plutonium. So there was one with plutonium-3, there were 2 with plutonium-4, and 1 with plutonium-6. And then they filled that up to the ground surface with sediment, again, from the native site. And they left them there for an extremely long period period of time. For 11 years for the plutonium-3 and plutonium-4 lysimeters, only 2 years for the plutonium-6, and I'll explain why in a minute. And they collected any of the groundwater or any of the rainwater that went through the lysimeter here at the bottom to see whether they were getting breakthrough of plutonium in the column. And then at the end of this time, they dug them up and cut them into sections and then analyzed what the plutonium distribution was in each as a function of depth. So this is a picture of where the lysimeters were. So they were actually buried in the ground down at the Savannah River site. And I'll just point out here, so each of these posts represents a different lysimeter. And I want to point out that there's grass growing on top of them because that'll be important in a moment. So this is what the results look like after they dug up the lysimeters and sectioned them off. So here we have depth in the lysimeter. So this is the surface, the ground surface, and right here is about where the filter paper was. And then the x-axis is normalized concentration. So here at 1 would be like 100%, and this is 99%. This is et cetera, et cetera. So you can see most of the plutonium stayed near the filter paper, where the filter paper was. But we did see some downward transport of plutonium, and this is you can be expected. You have water that's infiltrating through, and you would expect plutonium to go with the groundwater. Moving in a downward fashion. You can see here the blue circles in the line represent that plutonium-6, and you can see that it moved quite further and quite faster than the others, and that's why it was ultimately dug up. It actually had breakthrough of the column. So to avoid contaminating the local area, it was removed. But the others, you can see, we actually had some upward mobility of plutonium, and that was a little more surprising. It's definitely not what we expected. And so we think that some of this might have to do with the microbes that are in the area moving around, helping to cycle plutonium between plutonium-4 and plutonium-5. And then this part here, where we're basically getting straight upward mobilization of plutonium, we think is due to the grasses that were on top of the lysimeters. That basically, that's where the roots were coming down, and they were drawing water and therefore plutonium up. But that is still a topic of research right now. At the second version of the lysimeters. So this is what the new version looks like. I actually helped build this on one of my spring breaks. They call this thing a roll-off pan, which I guess is just a government term for a trash can, because that's what it is. It's a dumpster. And inside the dumpster, we've attached columns that are lysimeters to the sides. So each of these little tubes represents where a lysimeter is attached to the inside of that roll-off pan. And this is how we're collecting all of the effluent that comes through those columns, essentially. And then the inside of the roll-off pan is filled with concrete to make sure that they stay in place. So this is open to the elements, and the soil that's in there is soil from the Savannah River site. And they're meant to be extremely interchangeable. So if we're getting breakthrough, we can pull out a lysimeter and put a new one in. We can check lots of different types of sources. We can check, let grass or other plants grow on top of some lysimeters and not the other, so we can specifically look at that variable. It's just meant to be a very flexible system so that we can investigate lots of different variables and get lots of cool data to analyze out. So some of the research I did was looking at sediments from the Savannah River site to understand sort of this cycling of plutonium in the environment that might be causing some of this transport they were seeing in the lysimeters. So we took the sediments and expose them to a variety of different treatments to sort of understand what component of the soil might be controlling plutonium behavior. So we have the whole sediment. That means that we didn't do anything with it except suspend it in water. We have an ammonium oxalate treatment, and what this did was it targeted poorly crystalline iron 3 oxide minerals. So it removed them from the whole sediment. We have a hydroxylamine hydrochloride treatment, which targeted manganese minerals. So it made manganese 4 minerals that are typically insoluble. It reduced them to a more soluble manganese 2 that could then be removed from the suspension. And then we have 2 treatments which we call DCB. So DCB stands for dithionite citrate bicarbonate. So this is a 3-pronged approach at different for a variety of time periods and also different temperatures. And what these target are some of our other iron oxide minerals, goethite and hematite. So we basically did all these treatments to this sediment and then looked at plutonium, the interactions with these different solid phases to try to elucidate behavior. So there are a lot of studies where plutonium redox reactions are observed. Most of them are correlated to plutonium reduction. So if plutonium-5 is added to the system, it winds up as plutonium-4 associated on the surface. We call that surface-mediated reduction. And you can see that there's a variety of different minerals. We can have carbonate-based minerals. We have manganese-based minerals. We have iron-based minerals, silica, so just a whole variety. There are a few select studies where we see plutonium oxidation as well, but it's certainly not nearly as common as reduction behavior. So why might this be? We have 5 different hypotheses for this concept of surface-mediated reduction, which include radiolysis at the mineral surface, electron transfer due to iron or manganese in the mineral structure, disproportionation of plutonium-5, electron shuttling if you have a semiconducting mineral, and then the final one is a Nernstian favorability of sorbed species, which is a fancy way of saying there's a thermodynamic driver for absorption. So I'm going to go through each of these hypotheses to sort of explain them in more detail. So the conceptual model for radiolysis is that we have a solid phase suspended in the aqueous phase that contains plutonium, in this case in the +5 oxidation state, and plutonium, because it's radioactive, emits an alpha particle, and that alpha particle interacts with water to produce different radiolysis products such as hydrogen peroxide, which then interact with another plutonium in the aqueous phase and cause reduction, induce reduction to plutonium-4, and then that plutonium-4 sorbs to the solid phase. Electron transfer is something I'm sure you guys have come across in all of your chemistry classes. So this, say we have an iron 3 oxide mineral that has a little bit of iron 2 in it. The iron 2 can give up an electron to become iron 3, and that electron can then reduce plutonium-5 to plutonium-4. Disproportionation is essentially a reduction based on interaction with itself. There's no other external electron donors from the system, and it can happen both with plutonium-4 and with plutonium-5. So in the case of plutonium-4 disproportionation, we would need 3 plutonium-4 atoms to interact with each other, and that would yield 2 plutonium-3s and a plutonium-6. And these are the supporting reactions. There's a slow step and a fast step that give you this overall disproportionation reaction. The same thing can happen for plutonium-5 disproportionation. So you would need 2 plutonium-5s to interact to yield a plutonium-4 and a plutonium-6. So this figure here shows the fraction of plutonium in each of its oxidation states, 3, 4, 5, and 6 as a function of time in seconds. And you can see if we start with a solution that is 100% or completely plutonium-4, it's only a matter of time before we get this ingrowth of plutonium-3, 6, and 5. So this the hypothesis is, since this can happen with plutonium-5 as well, that you're just getting sort of an increased concentration of plutonium-5 at the mineral surface, which leads to this disproportionation, and therefore that's how you get plutonium-4. Because you can see from each of these reactions, according to Le Chatelier's principle, if you increase the plutonium concentration, you're just going to drive disproportionation forward. For semiconducting minerals, we can consider insulators, conductors, and semiconductors. So in an insulator, there's going to be a very large energy difference between the valence band and the conduction band. So you need a lot of energy input to the system in order to mobilize electrons. In conductors, there's actually an overlap between the valence band and the conduction band, but there aren't very many natural minerals that are pure conductors. But semiconductors, there is a gap between their valence band and conduction band, but it's a very small energy difference. And semiconductors, like a lot of the iron oxide minerals or manganese oxide minerals, the gap is low enough that even just exposure to light can provide enough energy to promote electrons from the valence to the conduction band. And then the electrons that make it into the conduction band can kind of move around the mineral surface to facilitate reduction of plutonium. And finally, we have this Nernstian favorability of plutonium-4 surface complexes. So this reaction here at pH 7, we can write the oxidation of plutonium-4 by oxygen as this reaction here. And then the log K, or the equilibrium constant, can be expressed here, where the AH2O represents the activity of water, and PO2 is the partial pressure of excuse me, of oxygen gas. So the log K for this reaction based on published values is 11.82. However, if we were to rewrite this equation to express it as the ratio of plutonium-5 to plutonium-4 as a function of the partial pressure of oxygen and the pH of the system, then we can calculate and then we assume that the ratio of plutonium-5 to plutonium-4 is 1. And we do that so that we can assume that there is some reduction of plutonium-5 to plutonium-4 at the surface. Then we can calculate a new log K value, and that would be 7.16. So then we can look at the difference, delta K, between those two equilibrium values, which comes out to be 4.66, and convert that into a delta E value, which turns out to be 0.28 volts. So this is just a really convoluted sort of back of the envelope calculation showing that really all we need is for the plutonium-5-4 redox couple to be increased by 0.28 volts in order for a plutonium-4 surface species to be more thermodynamically favorable than a +5 aqueous species. And so a lot of current research is going in to see how viable this actually is, or if we can measure changes in the redox potential of the system within that small margin. But it's not entirely outside the realm of possibility. There are lots of microbes, for example, out there that are able to change redox potentials within an order of magnitude of this value here. So those are the 5 hypotheses for surface-mediated reduction of plutonium. So I wanted to talk about a couple of the experimental techniques that I and my colleagues use in order to measure actinides and understand what's going on and how we're sort of attacking and looking at all of these hypotheses. And the 3 that I'm going to focus on today are batch absorption experiments, transmission electron microscopy, and X-ray absorption spectroscopy. So batch absorption experiments are sort of the bread and butter of my lab. Basically, you have a mineral that you suspend, usually in a low ionic strength solution, something like 0.1 molar sodium chloride or lower, and then you spike in your actinide element, and then as a function of time and pH, or choose your other variable, look at partitioning between the aqueous phase and the solid phase. So here's an example of some of my students' samples that are on this little rotator, and it spins end over end for as long as we need it to. And this is the kind of data that comes out. So we wind up making a graph of fraction sorbed. So this is how much of plutonium is associated with the solid phase as a function of pH in this case. And this is data for both plutonium added in its +5 oxidation state and in its +4 oxidation state on an iron oxide mineral called goethite. And so we can see that, so the open data points represent the plutonium-4 data, and the closed data points represent the plutonium-5 data. So we can see that we get distinctly different behavior depending upon the oxidation state of plutonium. Excuse me, we sort of get this really quick sorption of plutonium-4 and get almost complete sorption at around pH 5, whereas we get more of what we call an S-shaped curve with plutonium-5, where we have practically no sorption at low pH and then a very rapid rise in sorption behavior before we get near complete sorption under alkaline conditions. And a lot of this behavior has to do with correlating what's happening in the aqueous phase to what's happening with the solid phase. So in general, we have cations in the aqueous phase, and then the mineral surface is changing from a positively charged surface at low pH to a negatively charged surface at high pH. So you can imagine that we have electrostatic interactions going on here, particularly with plutonium-5, that a positively charged cation doesn't want to interact with a positively charged mineral surface, but at high pH, a positively charged cation is really interested in interacting with a negatively charged mineral surface. We can also use data like this as a function of time, and I don't have it to show you an example to show you today, but often we'll see that the curve for plutonium-5 over time winds up shifting and becoming more and more like that of plutonium-4. And so that's sort of an indirect indication that we are getting reduction of plutonium-5 to plutonium-4 in the system, because we're ultimately winding up with the same behavior as just a pure plutonium-4 system. So another technique we use is transmission electron microscopy. And so here we have just the standard electron microscope image of, in this case, quartz. Bet you didn't know that basically sand looks so funky under really high magnification. And we also use a Z contrast mode. So what this means is that the bright areas that you see have a higher mass, essentially. So we see hot spots on this silica that were not as easy to see just in SEM mode. And then we can zoom in on just small little particles and do some energy dispersive Xray analysis. So we can basically analyze just this little area and create a map for different elements. So I don't know how well it's showing up on your screen there, but there is a little bright spot, a red bright spot that correlates to this area here on this image, showing that there's a lot of plutonium right there. This is what the spectrum looks like that supports this image here. So this peak here is what the plutonium peak is, and the other peaks have to do with other elements that are in the system, such as silicon and oxygen. And then we can go to a high resolution transmission electron microscope to sort of zoom in even further to try to figure out what this looks like. In that case, when I was doing this work, I actually had to change microscopes. So I wasn't able to actually confirm that these structures here are plutonium, but based on some literature values or some literature data, we think that they are. But sort of this, grainy background would be the quartz particles, and these dark spots are plutonium. So we think that we're getting precipitation of plutonium in the system as a plutonium oxide, so PuO2 structure. And the final one technique that I wanted to talk about is X-ray absorption spectroscopy. And this is actually a catch-all term for two different specific techniques, the first being XANES. So XANES refers to X-ray absorption near-edge structure. And this is not something that I can do here at Notre Dame. It's not something that anybody can do at their university. We have to go to a synchrotron or X-ray facility, such as Argonne National Lab in Chicago, or these pictures in the studies I have done, we've flown out to the Stanford Synchrotron Radiation Light Source in California. So it all starts with this little tiny plate here, and you can see that there are 8 little grooves in here, and this is where I put basically a paste of the mineral that has been exposed to plutonium in this case. We put it in each of these little squares, and then we cover that with a special tape that is impervious to X-rays. And then that gets screwed into this larger plate, which again has more tape put onto it and is sealed so that a vacuum can be pulled on it. And then this is screwed on to a little piece of metal here. And this part is called a cryostat. And when it's on the instrument, we wind up filling it with liquid nitrogen. So this allows us to collect our spectra at really low temperatures. And that's important so that we know that any reduction of plutonium that we're seeing in our system is because that's what happens in the system and not because we were shooting a bunch of X-rays at it. So after we put our sample on the cryostat, it gets covered with another shield, and that's shown here. So this is what it looks like all assembled, and then that gets placed on the beamline. So I sort of show you all this just to show how careful we are when we do these types of measurements. There are multiple barriers of protection between us and our samples when we're running them. And even the beamline, there's an actual steel door between us, the X-rays, and our sample, and where we're sitting looking at the results on the computer. So at the beamline, this tube here, this is where the X-rays come in, and then our sample sits at a 45-degree angle between the incident X-rays and our detector. So this happens to be a 100-element germanium detector, which means every time we're getting a signal, we're getting it 100 times. So that means that we get really good counting statistics, which ultimately means that we can get relatively low detection limits with this instrument. So what does the data look like? This is what we wind up getting is a normalized spectrum. So we normalize the absorbance based on background and a standard so that we get a step height of 1, step height being 0 background before our edge, and 1 being sort of where all of these little waves happen. And this big peak here is what we call the white line peak. And the location of this peak tells us what the element is. So you can see, hopefully on this figure here, that as we increase the incident X-ray energy, we get different peaks that correspond to uranium and neptunium and plutonium. Usually we know what the element is that we're looking at, so we're looking at a much smaller energy range, but you could use it for elemental identification, I suppose. The exact energy location of this peak and its shape can tell us a lot about the oxidation state of our element. So you can see here that lower oxidation states tend to be shifted to lower energies, whereas higher oxidation states are shifted to higher energies, and that our actinides that are in higher oxidation states and have those axial oxygens wind up having a little shoulder to them. So the shape can tell us a lot about the oxidation state. So what we wind up doing is, in this particular setup, we added plutonium in different oxidation states to a system containing quartz, and then looked at the oxidation state distribution of plutonium on the surface. So we, in addition to running our mineral samples, we ran pure oxidation state analogs, or not analogs, but pure oxidation state samples, and then basically did a least squares analysis to determine what the oxidation state distribution was of plutonium on the surface. So this bar graph really summarizes the results pretty easily. It doesn't matter what oxidation state we add plutonium as, either plutonium-3, plutonium-4, or a mixed oxidized sample. The largest fraction of plutonium on the surface is plutonium-4. So it doesn't seem to matter what we do, we always wind up with plutonium-4 on the surface, which is a really interesting result that we don't really know why, and that's still something that both myself and a lot of other researchers are still looking at. So really what I've hoped I've driven home today is sort of why we need a fundamental understanding of actinide geochemistry, and that really requires understanding what's going on in solution and what's happening at the mineral surface in order happening with the mineral surface to get the interaction between the two. Then we can couple these results with what's happening with microbes in the area and hydrogeology in order to be able to predict actinide behavior, both in natural systems and in engineered systems. So with that, I'll leave you with a picture of my recent research group, and I'd be happy to answer any of your questions.
Okay. So remember, when you ask your questions, to raise your hand, say your name, so we all know. I think it's on.
Okay, cool. Hi, my name is
Hello.
Abraham Lee.
Speak loud.
Yes, anyways. Okay, my question is, whether, do you know any biochemical pathways that like bacteria could use with like uranium or something like that could be beneficial to the bacteria?
So I don't know how beneficial it is to the bacteria, but I do know that there are other research groups looking at microbial interactions with uranium and other actinide elements. It's not so much metabolized by the microbes. It's more of a mistaken identity type of thing, where they're searching for iron and instead they get uranium or plutonium. And I really think it just winds up serving as an electron donor for them, for some other processes that are going on in the microbes themselves.
Okay. My name is Cleve Hines. Okay. My name is Cleve Hines, and my question is, it seems that plutonium is so hard to dispose of, and it takes a long time to decay.
Mm-hmm.
Are you guys also working on an alternative to plutonium? Like research, you guys trying to find an alternative for it in use?
So plutonium is in the nuclear fuel cycle is produced as a function of irradiating uranium. So the uranium is what goes into a nuclear reactor, and you wind up getting a little bit of plutonium as a result of the fission process. So other countries use what we call it reprocessing. So they basically recycle these nuclear fuels so that they can use the plutonium again instead of having to dispose of it. That's something that we could do here in the US. It's just there's a large financial barrier to building a facility like that. But there are definitely some reactor designs that are looking at other sources like thorium, for example, or even looking at what are called breeder reactors. So they form the plutonium, but then are able to use that as fuel again so that it breaks it further down into less radioactive elements in the end.
Hello, my name is Irene, and you had mentioned that microbial organisms can be used to change the oxidative state of some of these more radioactive elements.
Mm-hmm.
And you also mentioned that we have radioactive waste buried underneath the earth. Is it possible to use the microbial organisms to change the metals underneath the earth so that they are less, I guess, destructive or dangerous?
So I suppose it's possible. We'd really need to look at sort of competition of, I'll just use plutonium as an example for a microbe over a mineral surface. Sort of the first issue might be is microbes move more than a mineral does. So they might actually wind up enhancing transport instead of degrading transport. But, you know, in the end, usually they're helping facilitate reduction, which is what we want, right? We want plutonium to be in that +4 oxidation state so that it stays immobile instead of in that +5 oxidation state where it just wants to move with the groundwater. As far as, you know, adding microbes to the environment sort of to augment it as an engineered system is probably not feasible due to the radiation field. Usually microbes aren't really happy and can actually be destroyed by the radiation itself if it's such a high field. But, you know, I have faith in a lot of the microbial people out there who study it, and I'm sure there is some strain out there that is extremely resistant to radiation that might be able to be used. So then you just have to convince the public that it's a good idea to basically inject non-native microorganisms into the ground, and that it will protect them. And that's sort of another barrier to overcome.
Hi, my name is Don. My question is, so on that one slide where you showed the plots from North Carolina in the ground with the jugs that you had filter paper, that's a fairly small distance that the plutonium is traveling over the length of your experiment. Is that transferable to the much larger and deeper distances that plutonium is buried when like in Nevada?
Well, that's a good question that's still under a lot of research, but I will say that there have been measurements of plutonium movement over a kilometer scale, both at the Nevada test site. So we've seen kilometer scale movement of plutonium, within the last, 20 or 30 years since we did nuclear weapons testing. And they've also made similar measurements in Russia, where they've had some plutonium spills there. So certainly it does transfer out to, a real field system as opposed to sort of a small scale simulated one.
Hello, my name is Dylan Zimmerman. So I worked in Boulder, Colorado this summer, and they had an incident about 2 years ago where one of their researchers actually dumped or spilled plutonium into a sink. And so my question is, when you have an incident like this, how do you get the plutonium out of the water? How are you assured that it's actually, is there a chemical way that you simply absorb it and pull it out, or you kind of just flush the system and hope it gets out of there eventually? Well,
I'm afraid my answer probably isn't going to be what you want to hear. I'm sure there are things we could do. You could put all of that water through an ion exchange column to be able to exchange out the plutonium. But in this case, it's probably more likely that they went with the old adage of dilution is the solution. I don't know that for sure. That's just what I would guess.
Don't quote you on that.
Yeah, don't quote me on that.
Hi, my name is Kadra. I know you mentioned that there is a small amount of radiation in bananas and, probably other fruit and things like that. But I was wondering if scientists use that to prove the hormetic model where there's a almost beneficial level that you can have of radiation? Is that something that they would use to prove that?
That's a good question I don't know the definitive answer to, but I think just the fact that we can eat bananas and not have cancer as a result of that probably, if it was going to prove any of those models, would prove the threshold model, that you'd have to eat really a whole lot of bananas in order to get cancer, versus that bananas are good for you. Which, they are because they're a fruit, but not because necessarily that they have potassium-40 in them.
Hi, my name is Helen Yu, and my question was, so other than isolation, are there any other ways to effectively reduce or remove the effects of the nuclear decay processes?
Unless you put it back into a reactor and essentially what it's called transmutation, so we turn it into another element, and unless you can keep doing that until it's a less toxic element, really isolation until you have 7 to 10 half-lives is our sort of benchmark for when something is considered gone, really is the only solution at this point in time.
Hi, my name's Dave, and I've got a question. Hi, yes, thank you. Yes, and at Chernobyl, there is a large amount of radioactivity that's actually in a complex biological environment. Do you see a difference in how plutonium oxidation occurs or reduction occurs in that complex environment there, that it's more than just bacteria, but it's entire complex life forms?
That's a good question. I don't know that I've seen a whole lot of studies looking at transport, at least in the subsurface environment, away from Chernobyl. A lot of the studies immediately after Chernobyl and even currently have more to do with atmospheric transport of fission products. So cesium is and iodine were the things people were really, really worried about. So like the amount of uranium and plutonium that's there, I think that they're making measures to ensure that it's not transported. And it's similar to some of the things that they're doing now at Fukushima Daiichi. You might have heard of this like ice wall and things that they're building to basically keep these isotopes in the immediate surroundings to prevent transport further off site. But as far as, you know, are we seeing really big differences in the behavior from like our more simplified systems that we're using now versus a real world field site? My answer is going to have to, I really don't know the answer there. The only sort of relevant thing that I am aware of would be the studies I mentioned earlier, where we've observed this kilometer scale transport of plutonium at nuclear weapons sites.
Hi, I'm Kelsey, and my question has to go back to goes back to the radiation. Nowadays, people are really worried about like cell phones and radiations that your cell phone gives off, especially like if you're charging it near your phone or if you're talking while it's plugged in. So, like on a scale of like bananas to, I don't know, to sitting in a reactor, how dangerous is the actual radiation coming from a cell phone?
So that winds up being a totally different realm entirely, because when they're talking about radiation in that concept, they're not talking about, you know, alpha particles or beta particles or gamma rays. They're talking about a different part of the electromagnetic spectrum. So unfortunately, I can't really answer that question, but I'm going to guess that it would be closer to the scale of bananas than sitting in a nuclear reactor.
Hi, I'm Danielle. I was just wondering what kind of material the lysimeters are made out of, and if the radiation from the plutonium would eventually penetrate it, or if it was meant to contain the radiation.
So the bottles themselves were made out of, it's basically a Nalgene bottle, like you guys would drink water out of. If you're hiking, or maybe in your labs there, you have big Nalgene bottles with deionized water in them for laboratory studies. So it's a plastic essentially that the lysimeters are made out of. So as far, I mean, if you're asking about like the alpha and beta coming off of the plutonium, certainly that and even the soil will attenuate and not pose a risk to anybody who might be in the area. But more importantly, I mean, the bottom of the lysimeters were connected to a tube so that nothing could escape the lysimeter. And they were collecting everything that came out of, all of the water that came out of the lysimeter to prevent contamination of the local area.
Okay, I want to thank you very much again for wonderful presentation.
Thank you very much.
I do have a question. Do you have opportunities during the summer for undergraduates to do research?
We absolutely do. If anybody's interested, I would recommend either emailing myself or one of my colleagues, Professor Burns also takes summer students, or even looking through our Center for Sustainable Energy runs a small program for undergraduates as well.
Okay, thank you very much.
You're welcome.